Welcome to part 3 of microfilaments, folks. So what we're going to do here is take a quick look at the general organization of actin filaments in cells, and then look at some more factors which regulate polymerization, depolymerization, and some proteins which control the organization of the filaments into networks. So cells that stick or adhere tightly to the underlying substrate, that's whatever they are attached to, have organized bundles called stress fibers. So this would be a stress fiber here, and it's a bundle of actin filaments. Rapidly moving cells, on the other hand, don't have such striking bundles. They don't have those stress fibers. If we look at the cell cortex, which is just below the plasma membrane, that has a network of actin which is cross-linked into a gel of microfilaments, and possibly has been best studied in red blood cells, and you can see that here. This would be the actin network in a lamellipodium, which would be part of the cell cortex. So lamellipodia are found in cells that crawl, and at their leading edge they have these thin filamentous projections called filopodium. So in the lamellipodium we have a branch network which is a little bit different from the gel we see in the cell cortex, and in the filopodia we find these parallel bundles, versus in stress fibers we have filaments opposing one another, and those bundles tend to be contractile in nature. So those are the kind of arrangements of actin filaments we can see in cells. Let's move on and take a look at some of the proteins which work on the actin filaments to promote polymerization and depolymerization. So cells can very carefully and very precisely control where actin assembles into microfilaments, and therefore it can also control the resulting network of actin filaments. To do this, they use a variety of actin binding proteins, just like we talked about with microtubule associated proteins. There's a whole bunch of actin binding proteins that control the polymerization and patterns of polymerization of actin into actin filaments. So we see control at the nucleation step, at the elongation step, and also we see an ability to sever the filaments, which in turn will stimulate depolymerization. So let's see what we got going on here in this diagram. It looks a little bit complicated. Now, as we talked about, if the concentration of ATP-bound G-actin is high, that is higher than the critical concentration for the plus end, then we will see polymerization into actin filaments. At some point, the actin concentration will drop below the critical concentration, and we will see polymerization ceasing. A lot of the G-actin in the cell, however, is not free and therefore not available to be polymerized, because it's bound by a protein called thymosin beta 4. And when actin is bound by thymosin beta 4, that means it's unable to be bound into the microfilament. Now, thymosin beta 4 competes with this protein called profilin, and so the competition between these two proteins determines, in conjunction with the critical concentration and the free concentration of actin, whether we get polymerization at the plus end. What I'm trying to do is build up a picture to show you that when you have more complexity, that's obviously a challenge for us to understand, but it means you have much finer control over these processes. So thymosin beta 4 and profilin compete for actin. What profilin does is it takes actin that's bound to ADP and switches it out for ATP. So you go from an ADP bound form, that's the D here, to an ATP bound form, and then profilin delivers the ATP bound actin to the plus end of the microfilament, and that's always in competition with the thymosin beta 4 protein, which is trying to grab hold of the beta actin and keep it sequestered so it cannot be polymerized on the plus end. At the minus end, we've got this protein called cofilin working. So that binds to and removes ADP actin from the minus end, which is going to promote depolymerization. That behavior can also stimulate severing, which again will promote depolymerization because it creates an additional minus end. So we've got these relationships here between these three proteins, and then we've also got to think about the critical concentration of actin and the free concentration of actin, and really that free concentration of actin is determined by the levels of actin in the cell, but also the abundance of thymosin beta-4 and profilin in the cell. So whether the microfilaments can actually grow is dependent on whether the ends are capped, and so there's a family of proteins called capping proteins, like this one here, CAP-Z, on the end of the plus end of the microfilament, and that will bind to the plus end and interfere with polymerization. We can also stop depolymerization by the use of proteins such as tropomodulin on the minus end. So the CAP-Z protein binds to the plus end and prevents addition of subunits there, and tropomodulin binds to the minus end and stops the loss of subunits there. So what we can do is regulate the abundance of CAP-Z and tropomodulin and their relatives, and that will help us stabilize microfilaments into structures such as stress fibers, for example, or the cortex that underlies the plasma membrane. So the cell also has this loose network of actin, and there are certain proteins such as filamin that you see here which can cross-link the actin filaments so that we actually get a network of bound together actin microfilaments. And so you can think about filamin here as kind of splicing these two actin filaments together and holding them in place to create this kind of network. So filament's a dimer. One part binds to one actin filament, the other part binds to another actin filament, and now they're stuck together. So that's going to kind of form a network. But then there's a whole bunch of proteins which can work to break up the network by severing and that kind of thing. So a protein called gelsolin can sever the actin filaments, and that will promote depolymerization. So that gives you a little bit more idea of the complexity that we see in the regulation of the filaments. I'll be back soon for part 4.